I'd like to just introduce our two speakers that are going to be tag teaming this particular session. So we have Dr. Carl Bradley. He is a professor and extension plant pathologist at the University of Kentucky. He works at the Research and Education Center in Princeton, Kentucky, which is in the western part of the state. He has statewide extension and research responsibility for disease management of soybean, small grain crops, and winter canola in Kentucky. His major focus has been on fungicide resistance and pathogens of soybean and wheat, and management of important diseases and pathogens that occur in Kentucky, namely Frogeye leaf spot and soybean cyst nematode in soybean, and fusarium head blight of small grains crops. We also have with us today Dr. Sam Markell. He is a professor and extension plant pathologist at NDSU. He's been at NDSU for 13 years now. His responsibilities are to develop and deliver management information for diseases of broadleaf crops grown in North Dakota. So these include sunflower, soybean, dry edible bean, canola, flax, and pulse crops. Dr. Markell's focus has been on applied disease management and monitoring pathogen population changes. And so we welcome you both and look forward to your presentation here today.
So maybe a bit of history about Carl and I that if you weren't on the talk yesterday, you probably wouldn't have heard. Carl was actually my predecessor. And he left about 14 years ago, right, Carl?
That's correct. I was the blip on the radar between Dr. Art Lamy and Dr. Sam Markell. And so Sam is in my former office, and it looks like he's cleaned it up quite a bit, as far as I can tell. And so thank you for the invitation. It's nice to be able to speak to you all. And one thing I'm missing, I wish we could have done this in Fargo. I would have really liked to gone back for those Holiday Inn chocolate chip cookies. I remember those being really good. So anyway, maybe you can send some to me, Sam. So I was asked to speak about Frogeye leaf spot. This is a disease that I actually started working on. When I left NDSU, I went to the University of Illinois, and I was there for about 8 years, and then came to University of Kentucky in 2015. And so I started my work in Illinois on Frogeye leaf spot and have continued that work. Before we get into it, I want to be sure that I do talk about some of the organizations that's funded this research. So a lot of the work that I'm going to reference was funded through the Soybean Checkoff, a lot of it through the United Soybean Board, more recently, some of it through the Kentucky Soybean Board. And then when I was at Illinois, the research that I initiated was all funded through the Illinois Soybean Association. And then I also want to give appreciation to many of the agrochemical companies and seed companies that have helped us out as well. Also want to mention, and I'll show you some things from the Crop Protection Network, but I want to mention this as a really good resource to get good multi-state publications on diseases. I know that Andrew mentioned this yesterday in his talk, and then Sam and I also mentioned it in our talk yesterday. So just want to reinforce that that's a really good place to go to get some information. Okay, as far as Frogeye leaf spot, I would say when I'm talking to the growers that I talk to in Kentucky, I really call this probably the most damaging foliar disease of soybean that occurs in the state that I live in now. And part of that is because of the conditions that we have in Kentucky during the summertime. We can be very humid. Those leaves can stay wet for an extended period of time, especially if there's there's a lot of hills and valleys, and so some of the fields are down in the valleys and trees surrounding those. They may stay wet until 2 o'clock in the afternoon. So anything that would keep those leaves moist for an extended period of time is pretty good for this fungus. And then warmer conditions as well. So warm, wet, humid, very important generally for this disease. So in regards to how this fungus infects, the fungus is Cercospora sogina that causes Frogeye leaf spot. It lives it survives in the debris, soybean debris. And then when you have rain that splashes down on that debris, there'll be these spores called conidia that'll splash up onto the leaves. So these spores don't move great distances. It's not like, southern rust of corn that might move miles and miles. These spores are just splash dispersed. So they're just going to move a little bit. So they'll be dispersed onto these leaves, and then cause an infection on the leaves, will cause lesions. And then within those lesions, there'll be more spores. And as it rains more, it'll splash more of those spores to adjacent plants and to additional leaves onto that plant. And so it does have this repeating cycle that can, cause more severity and more, new infections. We typically, here where I live now, at least, we typically don't see symptoms until after soybean plants start to flower. And part of that may just be because of when the inoculum is occurring and when we're starting to maybe warm up and have better conditions. I also think that part of that might be due to, that's typically when we start to see that canopy close where you can't really see between the rows anymore, is right around that time. And so that also provides maybe a better environment because of the closed canopy, not as much airflow that keeps those leaves a little, more moist for an extended period of time. And I stole this graphic, the disease cycle graphic, from this publication from the Crop Protection Network. So there is a good multi-state publication there that you can go to. Just looking at the symptoms here, typically those lesions or spots are going to be kind of a light tan to maybe a little darker tan color in the center of the lesion. And then around that lesion, they're usually going to be kind of a dark border, almost purplish in some cases. And these lesions can be circular and they can be very irregular in shape. So it's going to kind of vary on the shape that they occur in. When you have, pretty severe lesions that's been there for a while, the middle part of those lesions might drop out and kind of give these leaves sort of a shot hole effect. And what we see in Kentucky, and we can include Southern Illinois, Southern Indiana, states right around me, when we have conditions that are very favorable and we have a very susceptible variety, we might see really, really severe symptoms that are expressed like this. And so once those leaves have about maybe up to, starting at about 40% of the leaf area covered with these lesions, we may start to see those leaves start to wilt and curl. And then you can just kind of see the overall color of those plants. They're just almost a yellow. And so we see that they're very stressed when this happens. So we can have severity up to this. And then when it's really bad, you can really see those frog eyes occurring, like what you see in this picture. I took this picture when I was rating my trials last year, and I knew that I would be able to use it in a presentation. So I mentioned that the spores will form within the lesions. Normally, those are going to occur on the underside of the leaf, so you have to flip those over. And you can kind of see here, it's kind of hard. You can sort of see it with the naked eye, but if you have your phone with you or if you have a hand lens or something, you can certainly see them a lot better. I usually just use my phone nowadays and take a picture and zoom in. And you can kind of see the kind of the masses of spores that are within those lesions as I zoom in here. Kind of almost a grayish blue, steel blue kind of color of those spores. So that is one way to help diagnose what this is, if you are seeing those in the field. Typically, it has to be pretty humid, at least from my observations, to see those spores really develop in this fashion. And then here's some pictures that Sam had taken, and you can see that they look very similar. These are pictures from North Dakota, Sam. Do you know where you took this picture?
Yeah, this one was in Sargent County at a field. So, part of the reason we're talking about this is it's new to this state, really. Hadn't seen it before, and we saw it all over, and we'll show you a little bit more on the map. But this was in Sargent County, and this was pretty consistent in that particular field.
And then there's the underside, and you can kind of see with the naked eye, kind of that steel blue masses that are in those lesions. And then Sam took a really nice close-up picture, again, showing that mass of spores there. So apparently you can see the spores in North Dakota as well. It's been, I guess last year was pretty humid compared to maybe back when I lived there, it seems like, huh?
Yeah, it was hot and humid, and it was a lot easier to see these close to a shelter belt, as you might imagine.
Yeah. So a disease, and Sam, I don't know if you have this or not, but we have a disease that would occur in Illinois and in Kentucky as well, called Phylosticta leaf spot. And so it's sort of a look-alike disease that we see. And I guess I've got some other things here, but typically when we see this, it's usually earlier in the season. So it's before flowering, and you'll notice these dark kind of structure, these dark structures in here. Those are called pycnidia, and you won't see those with the frogeye leaf spot. And so I almost every year, like in June, I'll get people sending me pictures of this and thinking it's frogeye, and it's, you know, should I do something? And it's usually Phylosticta. Here's just another picture of Phylosticta. And at least under our conditions, I've never seen Phylosticta really be a concern. And so it's not something that we have to do anything about, but you can see here some of the pycnidia that'll form in there. And Sam, I don't know, I don't recall ever seeing that when I was in North Dakota, but
I've never seen it. Yeah, I've never seen it in North Dakota either. But it does occur to our south.
Okay, yeah. So something to pay attention to. You know, especially if you're seeing lesions really early in the season, be sure to check that. So every year, extension plant pathologists across the U.S. kind of come up with some numbers on how severe some of the diseases were each year, and we put some numbers to it as far as estimated losses for each of our states. And Sam and Dr. Berlin Nelson do that in North Dakota. I do it here in Kentucky. And so I just thought it was kind of interesting. I charted this out. I only went up to 2018. I guess I should update this slide, but I think it points out what I want to point out is that you can see that up until about, starting at about 2013, we started to see kind of a gradual increase of losses caused by this disease. And then it really skyrocketed in 2018. And so this has become a more important disease, not only sort of in the Mid-South area where I am now, or the lower Midwest, whatever you want to call it, but it's certainly spreading to other areas. I'm going to kind of show you the same thing here, just in a little different way. These are the percent loss estimates that we derive those same numbers from in the graph I just showed you. And this is from 2009. And I'm just circling the ones that are at least 1% loss. And so you can see Tennessee that year was 5%. Here's 2012, again, kind of where you might think it would be, sort of the Mid-South, Southeastern region of the U.S., where it's usually been a bigger problem. 2015, a little bit more widespread. You can see some of the Atlantic coast states there are involved, but then we jump up to 2018, and you can see that's really starting to affect a lot of northern states in 2018. You can see South Dakota and Iowa there with pretty decent losses considering the amount of soybeans that are grown in those states. And so, you know, we've kind of seen this trend, and kind of had the question, is if frogeye leaf spot is it now occurring more frequently in the northern areas, and should it be considered a consistent threat? And I think those are things, Sam, that I guess you will continue to learn in the upcoming few seasons, but there is a trend of seeing this further north than what we've seen before. So as far as management, some of your basic principles, as far as cultural practices, crop rotation can be a good way to help manage this disease. It is, you know, it does survive mostly on leaf debris and stem debris of soybeans. And so when you're rotating away from soybeans, that is going to help. In our area, we do have varieties that are resistant to frogeye leaf spot. They can be highly resistant, and they can kind of be moderately resistant. So there's a range of reactions we might see. There are some single genes that breeders can use to put into soybean varieties, and we find that one known as the RCS3 gene is very effective. Sam, I have no idea if any of those genes are in varieties in the maturity groups that you would grow in North Dakota, and so I don't know if you had any observations from last year or not.
Well, when we started looking for Frogeye, we saw it pretty much everywhere we looked. And if there is any resistance in those varieties, I would suggest that's not intentionally bred into our maturity groups, that it's just in the germplasm that was used as precursors for those. So I think that the answer is probably we don't know. I mean, we just don't know.
Yeah, and if you looked at seed company catalogs, I doubt that there would even be ratings for it, just because, as you said, they're probably not intentionally breeding for Frogeye leaf spot resistance in your area, just because of not really seeing it before, or at least not very consistently for that region.
Mm-hmm.
And then, of course, foliar fungicides, which a lot of my work in Illinois and Kentucky has been on. And so that's what a lot of the rest of the presentation will focus on. One of the things that got me really excited about this disease, when I went to Illinois, I started working on it and looking at fungicide resistance. And actually, what made me start doing this is my experiences at North Dakota. I worked on same crops that Sam works on. One of the crops I was working on was chickpea, and Ascochyta blight was and probably still is a big problem. Not very many fungicides that could control it, and Quadris was one of those. And we started looking at fungicide resistance and identified that that organism there was resistant strains to strobilurin fungicide. So I applied that knowledge I gained there to Illinois and started working on Frogeye leaf spot. And we found back in 2010 that the fungus Cercospora sogina we were finding strains that were resistant to strobilurin fungicides. And so those initial strains came from fields down in southeastern Illinois, western Kentucky, and western Tennessee in 2010. We continued to do the research. Additional states were doing some of their own work as well. This is kind of what it looked like up through 2013 in regards to resistance. 2016 and then 2019 here, and you can see how it's really spread into South Dakota and other areas. I didn't make a 2020 map, but for 2020, when I do update that map, we're going to include Wisconsin, and we're going to include North Dakota. Sam, you want to take it from here?
Yeah, so we first saw this show up, and it had not been officially confirmed or reported in the state until this summer. And so we first found it last week of August, and right away we recognized that this was a potential problem. And I need to credit one of my research specialists and a county agent. They were kind of the first that figured out what was going on. So we pretty much dropped everything. We contacted the North Dakota Soybean Council. They gave us some support to do some surveys as quick as we could. And you may remember that at least on the North Dakota side of the river, we had a hard freeze, and I want to say it was the 7th, 8th, 9th of September, somewhere in there. So that week of the freeze, right before the freeze, we went out and surveyed quite a few fields. So what you're looking at is a map of the counties we were in and surveying. The first number is the number of fields where we found Frogeye, and the second number is the number of fields in the county we surveyed. So basically from a line south of Trail County, it was almost 100% of the fields had Frogeye leaf spot in that. Carl, can I control this? No.
Yeah, is it working?
No, but maybe you could just advance this.
Okay, yeah, I'll do it.
But we did not get to all the counties, obviously. So the ones shaded here, I would suggest strongly that Frogeye was probably present in these counties. And I would also suggest that if you flip this map around, the other side of the Red River would look very, very similar. Frogeye was really, really common. And this was a surprise for a lot of reasons. First, that we had never had it before. But second, that it was so widespread. You know, often when you see a new disease show up, you're seeing it maybe in a couple patches or whatever. This thing was just absolutely everywhere. I want to advance that a little bit. We went out and we did a bunch of filming. We took a bunch of pictures. There's a video that's been used around a little bit to help explain Frogeye. And maybe the more important thing we did is we collected a ton of these samples during the survey. And so people, my graduate students and research specialists, everyone was out pulling leaves. And we sent hundreds of leaves down to you, Carl, to figure out if there was resistance to the strobilurin fungicides. That was the biggest thing we were looking for. Why don't you advance that, Carl? So unfortunately, but maybe not surprising, was that we did find the same resistance that Carl had identified in the Mid-South and into the Midwest. So how you read this map is we're in a field surveying it if there's a circle at all. An empty circle, like a few in Grand Forks and Trail County, means we did not find frogeye. The green is the percentage or proportion of those samples that were sensitive to the QI. So the headline, quadris, and the like, they still are effective. And the yellow are the ones that were resistant. So a couple things to point out here is that about a quarter of the samples, of the 300 and some samples we sent, were found to be resistant already to the strobilurin fungicides. And the second thing is that it's essentially everywhere, right? It's not located just in like Richland County or whatever. You see plenty of yellow percentages or proportions in almost every county. Actually, you do in every county that we surveyed. So it's pretty widespread.
Yeah, and so I'll just jump in here. So this is pretty common of what we would find in a lot of states, although there are a few more sensitive strains, I think, that we're finding in North Dakota. But the best way to change those where they would be all yellow is to apply just strobilurin fungicides, right? So that's a selection pressure that starts selecting out those individuals that are obviously already in the population. So if you're just spraying strobilurin fungicides, you're going to turn this map completely yellow. And those of you that probably work with sugar beets are well versed in the language of fungicide resistance and even with the related pathogen Cercospora leaf spot. The same principles certainly apply to soybean as well.
Yeah, and with this amount of resistance already in the population, it wouldn't take many single strobilurin applications to completely flip that.
Yeah, we've some of the work that we've done, we've actually seen that within a single season, that we can almost flip it to 100% within a single season. So something to definitely be careful about. Sam had asked me to touch on just the survival of Cercospora sogina, this fungus that causes frogeye leaf spot. And so there's been a couple of studies that have been done, and not nearly as far north as North Dakota, but we do know that Ann Dorrance's lab in Ohio, you can see Ohio there in a couple of counties where she did this work. Her student had leaves that they would put out on the soil surface, and looked at it over the winter of 2008 and found at both of those sites that fungus survived just fine. We did some work when I came to Illinois at 3 different locations. You can see there a couple in central Illinois, one down in southern Illinois. And we did this over 2 years. And from the 1 year, the first winter, we found that no matter if we buried the samples or had them on the soil surface, they survived just fine at all 3 of those locations. We left some out there and then went back 2 years later and found that really the only ones that survived very well were the ones in the southern Illinois location. So that might tell you something, Sam. Maybe 1 year of survival, but if you go maybe 2 years, it's maybe a little bit tougher. But you probably are going to have to do some of that work maybe to answer some of those questions for your geography.
Yeah, and I think we will. And I think if anything that I learned talking to Carl and looking at this is that the possibility certainly exists that this is going to survive the winter. I don't know if it's going to be 100% or 1%, but I think the possibility is definitely clear that we will, and I would say with great confidence until this week, which we're totally in the icebox now, but we will have some survival. We just don't know how much.
Yeah, and as you know, snow cover can certainly make a difference in a lot of these things. If it's insulated by snow, then probably going to have a better chance of surviving rather than just bare soil with, very, very cold temperatures. Not as likely, I would say.
Yeah, and we have a lot of bare soil out there, so we're crossing our fingers a bit. But yeah. I think the odds on bet would be that there'd be some survival. We just don't know how much.
I want to jump into some more of the fungicide control. And again, on the Crop Protection Network, there's a publication here called Fungicide Efficacy for Control of Soybean Foliar Diseases. And if you open that up, there's different groups of fungicides on the left and then different diseases up across the top. And I'm just going to zone in first on the Frogeye leaf spot column, which is where we're at here. And then over here on the left, you can see the frac group 11 or strobilurins or QOIs, whatever you want to call them. We have all those listed as P here, and that doesn't mean perfect. That means poor because of the strobilurin resistance that's very widespread. Looking at the triazoles, you can kind of see there's a range there from fair to very good. We do have one group in the NBC or benzimidazole fungicides, and that is thiophanate methyl. Some of you dry bean growers might know that as Topsin or some of the generic formulations of Topsin. We've actually found that is very good for frogeye leaf spot management, but that particular chemistry class has a very high risk for fungicide resistance. So that's not something that we would ever recommend applying on its own. But if you had it mixed with a good partner, a good triazole, then that would be a pretty effective product. And then we also have the new kind of this newer group, the SDHIs. And right now we only have one that's just a solo SDHI product, and that's Endura or active ingredient Boscalid. And it's actually poor for frogeye leaf spot control. And that's one interesting thing about these SDHIs is that the active ingredient really makes a difference. So they're not all SDHIs are equal on which pathogens they control. So we know that Boscalid or Endura is really good for white mold, Sclerotinia, but does not really have any efficacy against frogeye leaf spot. If you turn the page, you would see a lot more on here. And these are your mixed mode of action groups. I just kind of have several of the ones that are very good highlighted here. But here's just an example of Lucento is a mixture of a triazole and an SDHI. And so there are some SDHIs that do bring efficacy to the table there. And so there are some good products there. And here's another example here of this product called Acropolis, where we have that thiophanate methyl mixed with a triazole, tetraconazole in this case. So there are a lot of options here, and we do have some chemistry classes that are still effective. I just want to show you some data from some research that I've done in Kentucky looking at foliar fungicide management. We do a lot of things to try to promote disease in these fields. And so I always tell my growers, you're not a plant pathologist, don't do what I'm doing here. So the things that I do is I try to pick the most susceptible variety I can find. I don't rotate in these fields. I grow soybean on soybean, so there's a lot of that residue, a lot of inoculum. And then one thing that we do a lot of here in Kentucky is we plant no-till, and that's good for the soil. However, that's also good for plant pathologists in some regard, because that means that residue is on the soil surface, and then we know this is splash dispersed, this fungus is, so it's a lot easier for this disease to develop. And we make our fungicide applications at the R3 or beginning pod stage. We've looked at a lot of different timings, and at least for our area, that R3 is kind of the right timing for frogeye leaf spot. Now I know that white mold might be a concern for you in North Dakota, and that timing might be a little bit different. But what we've seen for frogeye, at least in our area, is that R3 stage is kind of what we're looking at. So this is across all those environments from 2017 to 2020. And you can see this is frogeye leaf spot severity. So the higher the bar, the more disease. This first bar is our non-treated check. The second bar is headline, which you probably know that that's a strobilurin fungicide. And so it's not looking too good here because of that fungicide resistance. And then we have a lot of these other products, and I'm going to bring in some of those chemistry classes. So here we kind of see that some of these are triazole plus a strobilurin mixture, but really the triazoles are doing all the heavy lifting. Here's some of those benzimidazoles. Here's Topsin on its own, but then Topsin mixed with the triazole and a couple of these different products, Froghorn and Acropolis. And then here's some of the newer products that bring in an SDHI. So a lot of these are mixtures of a triazole with a SDHI that are kind of doing the heavy lifting again, even though in some cases a strobilurin might be part of that. So we are getting pretty good efficacy, especially when we're using multiple chemistry classes that have still have efficacy against frogeye leaf spot. And when we look at yields, and these are all still in the same order here on the bottom, some of our best treatments, we got up to about a 20 bushel yield response in our area. I don't know if that's what you would see in North Dakota or not, but we can certainly see that with the level of severity that we have in our, mostly maturity group 4 varieties. And so generally where we had the most efficacy, that's where we saw the biggest yield response. So I guess in summary, that those QOIs or strobilurins, really widespread resistance. We're really finding that in most instances, those are not going to be effective in controlling frogeye leaf spot. And even in some of those areas that Sam showed that there's just a few resistant isolates out there, you apply just a single, just a strobilurin fungicide without a mixing partner in there, you're going to blow up that resistant population. So we have to be really careful still. Fungicide classes that we find widespread that are still effective in managing frogeye leaf spot would be your triazoles, your SDHIs, and then your benzimidazoles. And all of these groups do have somewhere between a medium to a high risk of fungicide resistance. So, for a while, all we had were kind of triazoles and strobilurins, and then the mixtures of those products. And I'm a little bit fearful for the triazoles because in my area, the triazoles have been doing all the heavy lifting. Strobilurins really haven't been effective for a number of years now. And so I think it's really important that we go beyond just a strobilurin triazole mix if you're really concerned about fungicide resistance, and be sure you have at least 2 effective fungicide classes. So that would mean 2 from this group here, just to help with fungicide resistance. And with that, some of you probably saw this picture yesterday. This is a picture that Marcia McMullen took of me when I was a young assistant professor almost 20 years ago at NDSU, probably looking at flax for the very first time, I would guess. And anyway, so I really enjoyed my experiences when I was at NDSU, and I'm glad that Sam took over. He's been doing a terrific job. And again, I was just a blip on the radar there. But thank you for your attention. And Angie, I guess we can do questions.